I want to introduce Margaret McFarlane-Guy, who is not only a remarkable scientist, but a close friend, so it's a very special occasion to welcome you to Harvard. Thank you for coming. Margaret got her education in the West Coast at UCLA, where she did her PhD, and then did a postdoc both there and a postdoc at the Scripps Institute of Oceanography at UCSD. Parenthetically, where I had done some years before that my PhD. So we know what wonderful San Diego can be like, especially around January and February. So she then took positions at USC and faculty positions at USC and at Hawaii. And for reasons that still mystify me, she took a position after that at University of Wisconsin at Madison. But just like the subject that she studies, she saw the light, went back to Hawaii a few years back, where she's now the director of the Pacific Biosciences Research Program. Margaret's interest has been for a long time on bioluminescence and the interactions that are occurring between microbes and animals that lead to light-generating animals, and has been a lifelong passion of hers that has generated really a remarkable amount of knowledge in these systems. She's been well beyond that study. She's taken the knowledge of symbiosis, but really spread it to organisms throughout the entire living systems. She's passionate about, even though she will tell you she's not a microbiologist, really an animal physiologist, but it really doesn't matter what label you put. She's been a spokesperson for the discipline that has had tremendous impact in the way many of us think and think about the future of the studies in the life sciences. She's received many, many honors, and I don't want to take time from her talk to enumerate them. Just will tell you she's a member of the Academy of the American Academy of Microbiology, of the American Academy of Arts and Sciences, and of course the National Academy of Sciences. But I think that if I had to ask Margaret what she's most proud of, and maybe I'm being presumptuous, is her passion for changing the way we educate people about the life sciences that drives a lot of what Margaret is doing these days. I believe that in a matter of a decade, we will look back and we'll say, who revolutionized the way we study and have integrated the study of life sciences? And I think we're going to point at Margaret. She's already done a wonderful pilot of this at teaching a course that really starts from the microbial world and teaches all of living systems at Caltech. And so, where she was a visiting professor under the Moore Fellowship. So without saying anything else, let me join in welcoming Margaret to this lecture. Thank you very much.
Thank you. Thank you, everyone. This is just such an honor to be able to come here and tell you about the things that I get excited about. And I want to thank Roberto for being such a kind host. He spent just about the entire day with me. I feel like I've taken up all of his time. But I really think that that being able to come here and talk to you about my work is really such a great honor. So what I'm going to do is, as the title suggests here, I'm going to talk about becoming invisible in the ocean. And so it's the story of the Hawaiian squid. So I'm going to talk today mainly about how this animal gets its symbionts from the surrounding ocean. It's been a big mystery that's kept me going for about 30 years on this system. Before I get started, this is the Kewalo Marine Lab at the University of Hawaii, where my lab is. With Diamond Head in the background, it's a really beautiful spot to do research. I've had the privilege of being there, both in my first time I was in Hawaii and now I'm back there. So let's get started. Okay, so this is my beautiful animal subject. This is Euprymna scolopes. And it's a very cute animal, very charismatic. And this particular animal isn't big. It's only a little tiny guy. Okay, so oftentimes my students will show pictures like this and not tell the audience how big, so they think it's like this thing, you know. But Euprymnus, they call them the dumpling squid. They're little tiny guys. And this animal has a lifelong relationship from the time it hatches. Within minutes to hours, it begins to engage its microbial partner in a lifelong relationship. And this is obviously a petroplate of Vibrio fischeri taken by its own light. And so these are luminous bacteria that form an association with the bobtail squid. Actually, the American name is the bobtail squid. The Australian name is the dumpling squid. Okay, so these are the stars of the show. And what I'm going to tell you about is I'm going to give you a little bit about their biology. So I'm not an ecologist or a behaviorist, but I'm going to tell you about that because it's really important to understanding how these guys work. Then I'm going to talk about why we study this association. Why has this been something that has kept us... Oh, okay. Is that good? Yeah. Why we've studied this association, why it's kept our attention for all these years. And then I'm going to go down deeply into how the partners recognize one another and make a partnership in the beginning. Okay. Euprymna is a night active predator. So it comes out down here at the bottom, that's me, collecting Euprymna. And so as soon as the sun comes down, they come out of the sand. So they bury in the sand during the day. They're not like other squids and octopus, which are free or in rocks or whatever. They actually bury in the sand. They bury so well that in the lab, if you looked into a tank that has one, you can't find them. They just bury in the sand during the day. And then they come out at night to forage in the water column. And so that's Ned Ruby, my partner over there, and this is me. And you just walk along, my students and postdocs and myself, we just walk along with a dip net and a flashlight in the evening. And every once in a while, you'll come upon some little bobtail squid going along, and you just scoop them up with a net and put them in a Ziploc bag with water, and you've got an animal for the lab. And they do very well in the lab. So what does this animal do with the light produced by these bacteria? So like I said, it comes out at night, and it's foraging in the water column. And if it didn't have luminescence, it would cast a shadow onto the sand. And this is a very common behavior in marine animals, is to use bioluminescence to do what they call counterillumination. In other words, what the animal does is it emits ventral luminescence of the same intensity, downwelling, the same intensity, the same color, and the same angular distribution as downwelling light. And in that way, it doesn't cast a shadow against the visual field of a predator looking up from below. So it's like a Klingon cloaking device or something. It protects itself from being seen by its predators. And there are predators out there. Those of you who have been to Hawaii, if you've seen the monk seals will eat Euprymna, little calabarito. And they're also lizardfish, and barracuda have also been found in the gut content. In gut contents of barracuda, you find the beaks of these little guys. So there are predators out there. So they go up in the water column and forage and use their luminescence in this behavior. So you need to have a few details of how their symbiosis is structured so that you understand in talking about it. So if you were to take this guy, anesthetize him, and dissect him open, what you would see is this is the inside of his body cavity. And actually, this is a her. I just realized her body cavity. This is bilobed light organ here. It looks like two kidneys stuck together. And this bilobed light organ is where the bacteria are housed. So they're housed deep inside of these tissues in 3 independent crypts. And these crypts are epithelia, and the Vibrio fischeri, this is a lower magnification histology section. And these are the animal cells, and the darkly staining things are populations of Vibrio fischeri, the symbiont. If you look at a little bit higher, a TEM mag, you see that the bacteria, which I've false colored here in purple, are residing along the apical surfaces of the host cells. With these, they're almost, the microvilli are so elaborate that the bacteria are almost completely enclosed in microvilli. So the interesting thing about this light organ is it's like a backwards eye. So instead of receiving light, it being, as the eye does, it emits light. But it has all of the same accessory structures to allow the animal to control where the light is going and how much is going, okay? Because the moon, the moon goes behind a cloud and things like that, and so it has to be able to change the amount of light going out. And you don't want the light to go out the other end. You want all the light to go out the ventral surface. So the light organ is like a backwards eye. It has, this is the photogenic tissue, the retina analog. Behind that is a reflector. And that reflector is made of imbricated layers of a protein called reflectin. And that particular reflector, it's like the back of a cat's eye. Any stray photons that leave the crypt spaces get reflected back out in this direction. So this would be ventral over here. That's where the light would be shining out. And then behind that is like a choroid analog that it's the diverticulum of the ink sac. So you can see that this is embedded in the ink sac of the squid. So this ink sac lines the entire back surface. So you're looking at the ventral surface here, in other words, the bottom of the light organ. Across the back, the whole thing is black. Now what you have here is you have diverticula that act like an iris that shut down across this culture. So a diverticulum from here will come over and act like an iris on the eye to occlude the amount of light going out. And then it has a lens that bends the light out. The thing that's remarkable is we've studied this at every level, from the morphology, the sort of anatomical relationships and the morphology, all the way down to the developmental biology and the biochemistry and molecular biology. This lens has the same protein, despite the fact that it's derived from muscle, it has the same proteins as the eye lens. So there's something very special about it. It's an aldehyde, for those of you who are biochemists, it's an aldehyde dehydrogenase derivative. But in order to be a lens, it downregulates all the muscle-specific proteins. So the biochemistry is just amazing. And the genes that are involved in eye determination, there are specific genes that when something's an embryo, it says this should be an eye, are the same genes that say this should be a light organ. So it's way cool. So that's where how the luminescence goes out. Okay, now my lab studied E. scolopes as an animal model of how, we now know that most, if not all, animals entertain long-term relationships with bacteria. How in the world do they do that? And so this animal is offered a really great model for understanding that. So the goal that we have in the lab, or have had in the lab and continue to, is to define the rules underlying mechanisms of symbiosis. And the idea is by studying something very simple. If you study something simple, you can probably understand it, whereas humans are extremely complex, and there are a lot of other really complex animals. To give you an example of the strategy, Nobel Prize awards in developmental biology are shown here. There were 6 Nobel Prizes between 1995 and 2007 in the field of developmental biology. And every single one of them was using an animal model system. And so they used C. elegans or the fruit fly, this is the worm, or the fruit fly or mice or the things I've shown across here. So this is a really important strategy that biologists use. And the really aggravating thing is that the head of the NIH, Francis Collins, hates models. And it's like, it's one of his life's mysteries. It's like, the Nobel Prizes go to models. What's the matter with you? But in the study of symbiosis, a whole bunch of different models have are now being developed, and this is the one that we work on. So what's the power of studying models for complex characters? Well, I'm showing 3 examples of complex characters here. A single egg, fertilized egg, goes from 1 cell to 10 to the 13th cells. And this, to me, is a minor miracle. George Clooney is a minor miracle. There was an egg that gave rise to George Clooney. But this is just an incredible thing that biologists have asked about how this happens since the beginning of biology, right? Another one is that there are 10 to the 14th synapses in the brain. And a lot of people are studying fruit flies and other small animals with complex neural circuits to understand how simple neural circuits work. The last one I'm showing here is the human body. We now know that the human body has something like 10 to the 13th cells, as I have here, and there are somewhere between 10 to the 13th and 10 to the 14th bacterial cells. So we are at least 50% and maybe up to 90% bacterial cells. So this is pretty remarkable. Now, think about the fact that most of this, this 10 to the 13th of the cells all have the same genetic makeup, as do what's going on here in the brain, these 10 to the 14th cells. But much of this, there are about 1,000 species of bacteria. I don't know. I asked Rob Knight recently, how many species are there? And he says, I can't give you a number. So it's somewhere hundreds to thousands of bacterial species in the human body, and they're all different genetic makeup. How complex can you get? So you turn to something, biologists turn to something fairly simple, like the Hawaiian bobtail squid. And as you know now, this is not a giant's hand. This is my technician's hand holding a little Euprymna. And what we use this animal for is to study the chronic colonization of animal epithelia. So all the surfaces of your body, the surface of your intestine, the surface of your eye, the surface of your lungs, and so on and so forth, all turns out seem to have a microbiota with which it co-evolved, each one of those surfaces. And so it's very complex, and so this animal is giving us an opportunity to say, if you only have one partner, what are they saying to one another? And it turns out that epithelial cell membranes are highly conserved. And what you'll see is that Vibrio fischeri interacts at 2 different stages in development with 2 different types of epithelial cells. One is ciliated, mucociliary, and this is Euprymna, and this is the bronchial epithelium of the human lung. And so these types of cells are highly conserved. So you expect that the conversation that animals have with bacteria is which is since the beginning of animal evolution, is going to be very highly conserved. And the other type of epithelial cell that it associates with is microvillus. And so the intestine is microvillus. And what Euprymna, Vibrio fischeri interacts with in the host squid is in epithelial cells that are microvillus persistently. Okay. So I want to just give you an example of what I mean by looking at something that is easy to access. So discovering the rules, an example. How does a particular region of an organism, so the example I'm going to give is your stomach. For example, the antrum of the stomach promote colonization by a particular bacterial type. So there is a microbe that for very many years we thought was a pathogen. And it is a pathogen, but Marty Blazer at NYU would tell you that this was part of the normal co-evolved microbiota as Helicobacter pylori. Post-reproductively, Helicobacter pylori will cause gastric cancer and all kinds of bad things and ulcers, right? And so because this generally happens post-reproductively, it isn't selected against. The carriage of Helicobacter isn't selected against. The interesting thing is that it turns out that if you carry Helicobacter, you have protection against getting certain allergies, and people who carry have a very low incidence of asthma. Sadly, we're losing it from, we carried at 90% of humans carried it at the turn of the 20th century, and we're down below 10% now. So we're losing, he wrote a book called Missing Microbes, which I highly recommend. Anyway, so you've got this thing. Now, in a human, think of how many environments these bacteria go down as they're making their way from the mouth down to the antrum of the stomach. That's a long way to go. If you're a bacterial cell that's 1 to 2 microns, that's a long, long distance. Well, they pass through many, many, many habitats. How do they know when to stop? How do they know when to attach? Do they, if they weren't ready to do that, do they start ready, or do they get ready by going through these environments, these previous environments? It's a very complex thing. So how does it get there? How does it remain there in a healthy state, are the questions. How many of you have seen this movie, My Dinner with Andre? This is like, you know, it's a generational thing. Okay, so My Dinner with Andre, it's a great movie. I love this movie. Okay, not a lot of action. For those of you who like action, this is not your movie. So these two guys come together for dinner, and when they first start talking to one another, you think you know who they are. By the end of the movie, you've changed your mind completely about who these guys are. Really, it's a very cool movie. But what is so compelling is that by listening to these two, you can understand their motivations with one another. You can understand exactly what their relationship is and exactly what's going on in their conversation. And I like to compare, this is like Euprymna vibrio fischeri. One host, one microbe, two players in this game. What I compare it to is the Kumbh Mela celebration, in which I think I have down there from the web, 110 million Hindus come together to celebrate Kumbh Mela. This is like your stomach or your intestine. I mean, there are, you know, how would you ever know what the conversations are, what the motivations are, what the relationships are among these? You can identify them. You can go in there and you can take a piece of DNA and say, that's this person, this person, this person. But getting to know how it works is going to be a very difficult thing. So the major questions that this field asks, that all of us in this field are asking, are most of us, most animals, do what they call horizontal transmission. That is, you pick up your symbionts every generation, and most of them, anew each generation. How do you do that? How do you harvest a rare symbiont from the environment? That's the first thing. How do you know? How do you recognize that? How do you make sure that over evolutionary time that there has been selection on determinants that allow you to recognize? So when you're born, you don't just take up an average sample of the environmental microbes. You take up and recognize very specific things. So that although they don't interact with you through embryogenesis, evolutionary selection has occurred so that you recognize the right guys at the right time. Development, the bacteria-induced brain development, your gut bacteria, we now know, influence your development. As a baby, you know, your brain develops properly only with your microbiota. Finally, stability. Once you get them, how do you keep them in a stable situation such that they don't overgrow the host or inflame the immune system, nor do you eliminate them, nor do you get rid of them by the activity of the immune system? A very delicate balance has to happen. And then finally, for most of microbiology, pathogenesis has been the way we thought germs. You know, we thought of our microbes as germs. What is the difference between when you've got a microbe in there that is one you've co-evolved with? What's the difference in the conversation that that microbe is having as opposed to a pathogen? And just to give an example of what we found in the squid vibrio system, all of these things that I list here, these are things that have been thought to be what we call virulence factors of the bacteria. So MAMPs. How many of you have heard of endotoxin? Endotoxic shock? Endotoxin is one of these MAMPs. And it turns out that in the squid vibrio system, these MAMPs behave to induce the development of the host animal. You know, it's something that's needed, required. And so all of these things I've listed here that are virulence factors of the bacteria, we have found in the squid system to be part of the normal microbiology. Host defenses, similarly, all of the things that I'm listing here that have been erstwhile associated with bacterial pathogenesis are found as part of the normal conversation. So for my money, these should not be called virulence factors and host defenses, but they're just animal microbe factors. They're animal microbe, they're the language that animal cells and bacterial cells use to talk to one another. So the analogy I make sometimes is it's not like French and German. It's the difference between an argument and a civil conversation. The same words can be used, it's just the way they're used that makes the difference. So, putting it in another way, just to reinforce this, horizontally transmitted symbiosis goes from establishment over the trajectory of the ontogeny of an individual, from establishment through development through stability. And what I'm going to do is I'm going to talk for much of the time now about how this happens in the beginning. Okay. In a horizontally transmitted symbiosis, the question is, how is the symbiont selected against a background of other environmental, potentially phylogenetically related, and potentially pathogens? Okay? So why use the squid vibrio system for this? Well, just a few more things to say about that. The size of the juvenile light organ is about 400 microns, or about a little less than half a millimeter across. So this is something that you can put in a confocal microscope and see the whole darn thing. I told you that the adults are about 2 or 3 centimeters as the animal. The babies are only a couple of millimeters long. And so they make a great subject for microscopy. The other thing is that the timing of colonization. So when we become colonized by our gut microbiota, they come into our gut, and within a few hours after being born, we have our first microbes in our intestines. I think they've done a study of how long it takes. It's about 3 hours. And that's a very similar time frame here. What's different is that in our guts, there's a consortium, and our gut goes through a very stereotypical succession. So what you have as an infant, you get your mature microbiota somewhere between the ages of 2 and 4. And after that, depending upon what kind of lifestyle you lead and what kind of things you eat, the ratios and proportions change, but the players typically stay not too different. Just to give you an idea of how complicated this can be, in a human baby, if the bug is going to the stomach, it's about 20 centimeters down the esophagus to the stomach. It's 20 centimeters or a little more. And getting to the intestines about 300 centimeters. Think of all of the environments that must happen. Vibrio fischeri, even though the light organ is 400 microns across, each crypt that it goes into is only about 100 microns across. And in that 100 microns, it goes through 6 completely biochemically distinct signaling pathways that change the way the bacteria are behaving. And so it's amazing, how much change there can be in such a little space. So imagine what goes on in the human body. Okay, the other thing is that the entire natural process can be studied in real time. And so that's what we do. So like I said, there are these long distances in the human, and I made a little analogy down here. In the human, it's like across the country is down to the esophagus, and then if you're doing this as an analogy with miles, or several times around the earth versus this being 1 mile. I mean, that's just gives you an idea of the difference in the distance that the organisms are traveling. Okay, so how does selection happen in this beautiful symbiosis? Well, embryogenesis produces tissues that promote harvesting of the symbiont. So during embryogenesis, they're in an egg. They're not interacting with the microbial world. The minute they hatch, they begin, well, actually, 10 seconds after they hatch, the cilia on the surface of these appendages on either side begin to beat. These appendages, I don't know if you remember back to the adult, these were not present on the adult light organ. These are juvenile specific, and they're for catching the right microbe. So if you look down here, what I'm showing is I'm showing water flowing in and out of the mantle cavity. So these are good squid. When they breathe, their mantle cavity goes in and out, and they pull water into the mantle cavity. Within the water, Vibrio fischeri is present. Very low concentration, but it's present. And so these features are really important. The symbionts aggregate on the surface, and then they move, then they sense a chemical, and I'm going to tell you how that sensing works. They sense a chemical. What you see here is red and green, red, the Vibrio fischeri are the red cells in here. This is the animal. These are the cilia are stained themselves. And then this blue is mucus that's all over the surface. So they just go down and into the pores on the surface. So what I'm showing here is I'm showing this landscape in a little greater detail. So each one of these has 3 pores at the base of these like arms of the ciliated appendages. And if you look inside, each one of these pores leads to an independent crypt. And so these crypts are sequentially developed during embryogenesis, such that there's a really big one, which is crypt 1 here. There's an intermediate size one, crypt 2, and a very small one, crypt 3. I've pulled down crypt 1 so that you can see it. It has, I told you about these 6 environments. There's an environment on the outside of the symbiote situation that's really important. Then there's the pore. And I should say, and I think I'm going to say in a minute, only Vibrio fischeri gets in there. So what happens beyond this point? So what happens is it goes down a duct. There's an antechamber that has all these antimicrobials in it. And then it goes down and resides permanently in these crypt spaces. Okay, this is the journey. If you were to do a cross-section of the organ, I say it's an exclusive environment. In the absence of Vibrio fischeri in the water, nothing else colonizes. So at that pore, it goes up this long duct. There's an antechamber here that you can't see, and then it goes to a bottleneck, and they swim into these crypts, and that's where they reside for the life of the animal. Now, something I'm not going to talk about today is that this is on a really profound circadian rhythm. At dawn, when they bury in the sand every morning, they let loose 90% of their bacteria into the surrounding seawater. And they leave a little patina on the edge of this crypt space that grows up to fill the crypts so that when it comes out at night, it'll have a full complement of bacteria. So every single day, beginning the first day of dawn, it vents 90% of its symbionts in the surrounding seawater. But it interacts when it's growing and whatnot with 2 cell types, this epithelium and this set of chemocytes. And these are blood cells. They're the antimicrobial macrophages of this animal. And they sample the crypt space and bring the information about what's going on here back to the circulation. The entire body of this animal knows that these bacteria are here. I have a graduate student in my lab looking at the gills and the eye and whatnot. When you colonize this crypt space, gene expression changes in the eye of the squid. Gene expression changes in the gills of the squid. This entire body knows that there are symbionts there. Okay. What's interesting is that, I showed you that little bottleneck just before the crypts. Well, what happens is, if you give them a lot of Vibrio fischeri in the surrounding seawater, they'll make a big bunch of them. That big bunch will go into the pores up the antechamber, and they'll get to the bottleneck. On average, a single Vibrio fischeri cell goes in through that bottleneck and grows out. And all those other guys are left behind. They get killed. So it's like a block to polyspermy or something that's going on at that bottleneck. So on average, a single bacterial cell. So if you take an adult from out in the field and you ask how many strains of Vibrio fischeri it has in it, it will never have more than 6 because it has 6 crypts. Okay? Okay, so this is what you can see if you're studying this. So what this student did, and this was one of the ways that we proved that a single type is, so we put in red fluorescent bacteria. We gave the squid a mixture of red fluorescent bacteria and green fluorescent bacteria. And we asked what colonized what. And you can see that 2 of the crypts got colonized by red bacteria, and 1 of the crypts got colonized by green bacteria. So you can see that this was a case where you only, they're very well separated from one another. So how does this all happen? Take one step back. Let's begin in considering the choice of partners. If you get the right partnership, you're going to have a great time, just like Laurel and Hardy here. So I should start this by saying, as Roberto said, I'm actually an animal physiologist biochemist. And I have had the privilege of working with Ned Ruby, a microbiologist. He studies the microbiology side of this symbiosis. And very early on, I wanted to do everything natural. I wanted to put the babies into mother's water, and get them colonized naturally, the way they would naturally get colonized. And Ned said to me, oh, no. You're not doing that. This is going to be all controlled. We're going to use one strain. We're going to use ES114, which was one of the strains that he isolated. So for 28 years, we used ES114, thinking that this was the way it worked. Okay, so I'm going to tell you about what we found in the last 2 years. So it turns out there are 2 distinct populations of Euprimna. And early on, we found that there are 2 haplotypes of the host. One living in, this is obviously the island of Oahu in Hawaii, and this is Kaneohe Bay. Kaneohe Bay is a weird place. There's not a lot of, even though it looks like there's a sandbar here, and there's not a lot of exchange. So there's a lot of weird evolution that's gone on here. And one of the things is that this population of Euprimna has been fairly well isolated. So there's a population here, and there's a population here. And for those of you who are Drosophila biologists, when we did the CO1 sequences and whatnot, the Drosophila biologist said if this were a Drosophila, they'd be different species. So they're different enough, okay? So we asked the question of whether or not, if you're a squid from the other side of the island, maybe you have different bacteria in your light organ, different strains. Well, it turns out the answer to that is no. The strains from either host population chose both populations of bacteria equally. The Kaneohe Bay, so down here I have MB and KB. MB is Mauna Lua Bay, and KB is Kaneohe Bay. So they chose, the animal didn't care at all about where the strain came from. And so this was a very talented postdoc in Ned's lab, Clotilde Bongrand, who's been working on this. So there's no difference there. However, what she did discover by doing these competitions with the 2 strains, she said, well, maybe they're not different, but they like the Mauna Lua Bay ones, like the guys from Mauna Lua Bay, better. And so she put them head to head. She didn't find that they liked the guy from their own place, but she did find that there were some strains that would always dominate in the light organ. In other words, they would be all 6 crypts. All 6 crypts would be colonized by what she called the dominant strain. And then there were some strains that she called the sharing strains. And that is, they would get in and they would share the light organ. In other words, there would be more than one type strain. You could get up to 6 different strains in there. doing this, she was able to create this dominance hierarchy. And so she has these D-type strains that get in, they just fill the crypts. They're very dominant. And she could create a hierarchy of who was most dominant by putting these groups of these together. These sharing strains that she called the sharing strains. Now notice, this is ES114. So ES114 is a sharing strain. And so all this time, we've been studying this guy down here. Okay, so how does this work? The question is, why in the world don't the dominant strains sweep? Since they're so fast and they're so good at colonizing, why don't they just sweep the whole population? Why do you have sharing strains? Well, it seems like in culture, the sharing strains do better in competition. So perhaps out in the environment, they persist, and actually she did some experiments to show that they persist longer when they're in the environment. The sharing strains do. In Squido, the dominant strains dominate. So this is obviously a baby squid, and this is a baby squid light organ. Okay. So maybe that's what's going on. So then we asked, I wanted to mention that if you did the phylogenetic relationships among these, now mind you, this is a sharing strain, and this is a sharing strain, and this is a sharing strain. This guy is from Mauna Bay, and this guy's from Kaneohe Bay. Okay, so Kaneohe Bay and Mauna Bay are mixed up here, and Kaneohe Bay and Mauna Bay are mixed up here. These are the sharing strains, and these are the dominant strains. So they're completely different, completely different. Okay. So what's the difference? So we sequenced all of these with Ed DeLong and Martin Poltz, and this is the difference. So the dominant strains here have 4.5 kb, and the sharing strains have 2.5 kb. Excuse me, 4.25 kb. So the sharing strains are missing 250 kb of DNA that the dominant strains have. So we got all excited. We thought, it's going to be a pathogenicity island. We'll just get rid of that. It turns out that 250 kb is shot all over the genome. And if there's a microbiologist in the audience that can tell me how this happened, I would love to hear it. But it's really an amazing thing. It's shot all over the genome. And so we now have a method for pulling out all of these genes. And we're going to be doing that to see which ones are conferring specificity and so on and so forth. But it's just an amazing thing how different they are. But we now know, it's for a long time, studying human biology, we knew that there were 1,000 or so different bacteria, hundreds to thousands. But we now know that there are tens of thousands of strains. And so what is the impact of strain variation? Why do we maintain different strains? Do they have different habitats? Do they have different strategies? And that's the kind of thing we're getting at here. How is it that this variation is maintained? It must be important to the biology. So then we asked the question, we can watch this happening, as I mentioned. Where does the dominance happen? How does it happen? Well, after I talk to you about this, I'm going to tell you a little bit more about ES114, because that's where we know the most about how these animals get their symbionts. But I'm not going to be able to tell you about the dominant strains, because they're doing everything differently. And I'm going to tell you how it works. So the journey of the dominance, where does it exert dominance? Right off the bat. So the bacteria aggregate at the beginning. They're pulled out of the seawater. And this is a biomechanical problem. By 2 hours post-inoculation is when it happens. This is ES114. What happens is, I'm going to show you, ES114 just sits there in an aggregate, and then it goes into the pores. That's the social strain. See that little green thing? Is Vibrio fischeri. There are about 5 cells there when you give them what they typically get. This is the dominant strain. When we got this under the control, it was like, oh my God, they are just sucking Vibrio fischeri out of the environment. I mean, they have, it's just amazing. Not only that, I'm going to show you that ES114 takes a few hours to do everything it needs to do to invade host tissue. These guys go in within 15, 20 minutes. So there's something really remarkably different. So I'm going to talk about ES114, and I'm going to talk about the biochemical and biophysical determinants. So keep in mind that Vibrio fischeri is about 0.01% of the bacteria in the surrounding seawater. These animals are a couple of millimeters. They have this problem, very low Reynolds numbers. They have to pull in these bacteria and engage them against really big odds. So as I mentioned, it occurs outside on the light organ surface. You can see this is a RFP labeled bacteria above a pore. And so I'm just going to spend a couple minutes talking about the biophysics because I understand Eva Kanso was here recently and she talked about this. So this is a collaboration talking about associating what are the biophysics of these cilia. And this was a collaboration done with Caltech and USC. So this is John DeBerry, who's now moved from Caltech to Stanford. And Jana Nauroth, who's at Emulate here. She was at Wyss Harvard School for Bioinspired Design for a while, but she's sort of the central person in this. Eva Kanso was the mathematical genius. Scott Fraser was the imager, and Ned and I were just looking on, basically, really. But we asked one of the only question I'm going to tell you about, and the only thing really relevant to my argument here, is what we did was Jana looked and she asked whether or not these cilia size select. And what she did was she put in 2 and 4 micron particles. And the 2 micron particles are green, the 4 micron particles are magenta. And Vibrio fischeri is 2 microns. Is the animal able to distinguish? So where you want to be looking is you want to be looking right in this region here. Okay? Every once in a while it catches a magenta one. But at the end of the day, by and large, it's able to sieve out the 2 micron particles. So it's able to sieve out those particles in the surrounding seawater that are the size of Vibrio fischeri. Well, any microbiologist in the audience will tell you, well, every other bacterium out there is 2 microns. So this can't be all that there is. And indeed, there's biochemistry here. So what happens is, at first, the very first thing within 60 minutes, they respond to peptidoglycan, other bacterial cell walls, just a general molecule that bacteria shed, by shedding copious amounts of mucus. This is Spencer Nyholm, a former graduate student who's now a professor at UConn. But they shed copious amounts of mucus on the surface, and that's the platform where they aggregate. So in this backdrop, while Vibrio fischeri ES114 is gathering, I had this phenomenal student who asked, can the animal sense Vibrio fischeri against a background of 1 million other bacteria? So what she did, she showed that host genes are regulated specifically to the 5 Vibrio fischeri ES114 that attach against a background of thousands of other cells that are nonspecific. And they attach to only a couple of cells on the surface of the animal, and the light organ is 10,000 cells. So there's a wave of gene expression that goes across the organ in response to Vibrio fischeri. She compared hatchling and aposymbiotic, that is no Vibrio fischeri, but 1 million other bacteria, against Vibrio fischeri, and she found a lot of genes were regulated. We did this with Phil Rosenstiel at Christian Albrecht. So what did she find that was important to the issue? She found that what Vibrio fischeri does with this chemistry is it winnows, what the squid does is it winnows to Vibrio fischeri. The bacteria attach. They say to the host, Vibrio fischeri is here, change your gene expression. That gene expression changes, and it changes the game. It changes the game in favor of the specific symbiont. So what it is it pours out a bunch of antimicrobials in very low concentration. So these antimicrobials are in concentration so low that all Vibrio fischeri does is respond to them. It gets primed. They get primed. Because the ducts and antechambers of the light organ, which as you recall are specific to Vibrio fischeri, are on fire with these things. So they get primed. So priming is being prepared for the next steps. And so one of the things that they do is there's a blood pigment, hemocyanin, that gets clipped into an antimicrobial. So these are the ones we've proven by various experiments and with various mutants of Vibrio fischeri are important. And chitinase. They get primed to chemotax to chitinase. And so their specificity is determined, they get primed, and then they get primed to chemotax into the tissue. They have to know where they're going to go, right? So knowing where to go with chitobiose. So what happens here is that there's a lot of chitin, polymeric chitin, in the mucus. The bacteria attach. They tell the host to change the expression of a chitinase. This chitinase, that chitinase clips that chitin into chitobiose, which primes the bacteria to chemotax to chitobiose. So what has priming done? I mean, what can you see that priming has done for the situation? Well, if you look at bacteria going into the duct, you'll see that there is a mixture of what you would see on TEM. This is a transmission electron micrograph, obviously. And you see this group in here? They look stressed. They look melanized, which is there's material that's laid down on them that is very stressful. So these bacteria look kind of stressed. These bacteria look like they're doing okay here. Okay. What we did was we labeled the bacteria. We labeled Vibrio fischeri in collaboration with Anders Maibaum at the EPFL. We labeled the bacteria and did a technique called nanosims. So what you do is you give the bacteria a substrate that has N15, nitrogen 15 and carbon 13, heavy isotopes. And it labels them so that you can see those isotopes in a microscope. So you thin section them, and it's a combination of microscopy and mass spec. But look at this. These healthy bacteria are all the Vibrio fischeri. So we label the Vibrio fischeri here. And these are the guys that are dead. So it's been a very effective selection mechanism right up there at that duct. And we're doing that as well. The question is, do we need priming? And we know that pathogens need priming. There are lots of pathogens that people have studied that need priming to be able to respond with an appropriate pathogenesis. Do our normal microbiota need to be primed to land in the right place? And do we need to have our chemistry correct for that? Oops. So the last thing that I wanted to mention is I started off with talking about this light organ and what it did for the squid. The symbionts defective in light production do not persist. So if you put the luminous bacteria in this, a defective mutant that doesn't, we call them delta lux. If you put a delta lux mutant in the surrounding seawater, the animal takes it up just fine, right away. All the things I've talked to you about that the animal does, it does with a dark mutant. It gets in there and it doesn't do something, it's not doing its job, right? It just is dim. And the animal says, oh, wait a minute, you're not doing your job. And within 48 hours, they're down to 50%, and they're just eliminated from the light organ. The fascinating thing is if you put a mutant that only colonizes to a low level, but is luminous, they'll stay in there just fine. And they'll make light, and you can see all that stuff. If you put a wild-type bacterium in with the mutant bacteria at 50-50, they're not complemented. You would think light, so the delta lux mutants are not complemented by the presence of a dark And so the answer is almost certainly that, that they're eliminated, that they don't do as well. They only get into one crypt, right? I mentioned that a single one gets into a crypt and it just doesn't make it. It's not that they're together, and that's why they're not complementing. Okay. So this is just to show the ducks and the crypts, and actually they're confined to the crypts during the day. So the light organ, as I mentioned, expresses proteins that allow the light to see, the animal to see light. Luminescence is required to induce normal development. So not only does the animal kick it out, but light production, which is only required early on for early early development, it's only required at the beginning. When the bacteria are there, the light is critical. And the last thing that I didn't talk about, I just mentioned it briefly, is circadian rhythms. This symbiosis is on a profound circadian rhythm. And the bacteria drive the circadian rhythm. And if the bacteria are not present, that rhythm does not go. And we know that the bacteria require light. They must be making light to induce this rhythm. So the light is very profound, has a very profound effect. And I have this student that's doing the light production with the transcriptome. 2/3 of the light, if you compare a delta lux for gene expression with a wild type, how many genes they induce, 2/3 of the change in gene expression is the light production. It's phenomenal. So in conclusion then, I hope I've shown you that animal models of symbiosis promise to provide the rules of engagement. And specificity involves both biomechanical as well as biochemical factors. Light production is absolutely essential to the whole thing, from the biochemistry to the development to the behavior requires light. And imaging is a powerful teacher, and something that we can use. So I've shown you what goes on in this ridiculous little squid that has one partner, just one partner. All the things that it's done have been selected for over evolutionary time to make sure that it works the right way. Think of what has to go on here. It's just remarkable. And it's so much fun to work with the people who are doing this kind of biology, because we can talk back and forth. In fact, as I mentioned, MAMPS, induced development, we did that early on, and the Pasteur Institute people found that the exact same molecules that drive development, the bacterial molecules that drive development of the host, drive the development of the mammalian intestine. And so these are very, very highly conserved things. So I'd like to say thank you to my partner, Ned Ruby, of course. This was the 25th anniversary at Madison. This June, we celebrate the 30th at Scripps Oceanography, where I was a postdoc and I started my first looking at squids. It's been such a great time. These are graduate students and postdocs who could make it. There are now, I'm not a good counter, there's something like 16 or so labs that work on the squid Vibrio system. So down here have been the generous people to give us money to keep our habit. And the other thing that I need to do is I need to acknowledge the laboratories with whom I've worked. 33 labs at 23 institutions in 4 countries. Every time we wanted to do something, you'd open a door and you'd go, oh my God, apoptosis. What do I know about that? And then you'd have to learn a lot about it, but there were always people who were willing to collaborate. So with that, I'm going to shut down the lights, and I wonder if I can shut them all down. Can I shut them all down? No? And just say, Mahalo nui loa.